Okay, so let's talk about how gene expression is tightly regulated in eukaryotic cells. And we're going to focus on control mechanisms surrounding transcription, mRNA processing, translation, and protein activation. So, even though the basic principles of transcription are very similar between prokaryotes and eukaryotes, like I mentioned previously, eukaryotic gene expression is much more complex. So one reason is that in eukaryotes, each gene is going to have more regulatory proteins. So this does allow for more precise control, but it also introduces a higher risk of something going wrong. So a key group of these proteins are what we refer to as transcription factors. And these are just DNA binding proteins that are going to help the RNA polymerase bind to the promoter region of the gene. Several transcription factors are actually needed for each gene to be properly transcribed. And they don't just bind randomly, they actually form a complex that's going to help unwind the DNA helix and help position that RNA polymerase correctly. And these factors can be reused in different combinations in order to control multiple genes. This also means that even if one transcription factor is defective, the consequences can be severe because they all accumulate and act together. And mutations in transcription factor genes are associated with a lot of diseases, one of which is Huntington's disease, which we did talk about previously. So some of these transcription factors can bind to the enhancer regions of DNA, often far from the gene that they actually regulate. And they're going to speed up transcription by stabilizing the transcription complex. So here's just an illustration of what I was just talking about. So they can speed up the transcription, again, by stabilizing the transcription complex. So just one transcription factor can actually dramatically alter the cell fate. And one really good example is MyoD, which can activate the entire genetic program that converts fibroblasts into muscle cells. Similarly, we have the eye transcription factors in fruit flies, and these can initiate the development of a complete eye, even in places that don't usually grow eyes, if it was activated. So going back, once transcription was complete, we had our newly made pre-mRNA that had to undergo processing before it could leave the nucleus. And we talked about how we need to remove those introns, which are the non-coding regions, and then splice together exons, which were our coding regions. So eukaryotic cells can use something called alternative splicing, and this allows them to produce different versions of a protein from the same gene. And what this does is it increases protein diversity substantially. So for example, the DS-CAM gene in fruit flies, it can generate over 3,800 different protein combinations just from alternative mRNA splicing. So once that mature mRNA reaches the cytoplasm, we have additional layers of control that can come into play. So whether or not translation actually happens is actually going to depend on cytoplasmic proteins and the environmental conditions. So for example, red blood cells won't produce hemoglobin unless heme, the pigment, is available, even if the mRNA for hemoglobin is present. Also, the longer an mRNA molecule survives in the cytoplasm, the more protein it can produce. So the stability of the mRNA can be influenced by the length of that poly-A tail that we talked about in the pre-mRNA processing. It can also be influenced by the presence of hormones. So after a protein is translated, it's not always immediately active. So a lot of proteins are going to require post-translational modifications in order to become functional. So a good example here is insulin. Insulin is synthesized as an inactive precursor, and it has to be processed before it can become active. So this kind of delay is going to allow the cell to kind of hold off on protein activity until it's actually needed. Okay, last thing I promise for this chapter is that in multicellular organisms, cells are not working in isolation. They are constantly communicating through chemical signals, and these signals are going to influence each of the cell's behavior. So during development, these signals can help determine what kind of cell a stem cell will become. And then later in life, they can regulate everything from tissue repair to metabolic activity. So they work through something called cell signaling pathways. So we have a signaling molecule that's going to bind to a receptor on a target cell and trigger something called a signal transduction pathway inside of the cell. And it's actually not the original signal molecule that brings about the change. It's actually a cascade of internal events that's going to follow. And ultimately, it's going to affect gene expression or protein function.